Welcome back, guys. This is part 3 in our section covering biological molecules and polymers. In the first section, we talked about carbohydrates. In the second video, we talked about lipids. And in part 3, we're going to talk about proteins, which are made of amino acids. So this sort of generic molecule is showing you the setup for one amino acid. Amino acids are the monomer that's used to build proteins, or sometimes you'll hear them called polypeptides, which are your polymers. And the general pattern that we've got here is a backbone that never changes. So this entire structure is the backbone of an amino acid. On one end, we have an NH2, which is an amino group. On the other end, we have a carboxyl group with this is carbon, double bound to an oxygen, and then single bonded to an OH. And in the center, this alpha carbon, which is always the one in the middle, is bound to a hydrogen on one side and something called an R group on the other side. In all of this, the R group, which is also called the side chain, this is the only part that's actually variable. The rest of it is entirely the same all the time. And so whenever you see amino acids and proteins being written out in sequence format, you're always going to see this pattern, NCC, NCC, over and over and over again. The only thing that's going to change is which R group or side chain is coming off of it. All right. The other thing about amino acids that's a bit different than carbohydrates and lipids is we have a lot more variety. So in this table, this is showing you the 20 amino acids that are the most common. These are the 20 that are used in humans. There are some other variants that you'll find in bacteria, but they're not very commonly discussed. So it's unlikely that you have to memorize all of these, but you should be able to look at these structures and at least tell that they are an amino acid. And if you notice, the backbone, right, nitrogen, carbon, carbon, always the same. Doesn't matter which amino acid we're looking at, that part is identical. So whether it's glycine, where the R group is just a single hydrogen, right, all the way up to these very large, complicated structures, some of them that have rings and side chains with all sorts of different properties. The R group is highlighted in blue here. Again, that's the variable portion. And all you should really probably know from this for right now is the R group is what gives an amino acid its distinct properties. Okay, and so some of these are nonpolar, some of them are polar but don't have a charge, some of them do have a charge but it's positive, some have a negative charge, and you have all of these different varieties so that you can assemble amino acids into proteins in an almost infinite number of combinations. Since you have 20 different possibilities, we're talking about trillions of different combinations of amino acids that you could use to make a protein if that protein is going to be several hundred amino acids long, which is typical, right, for a protein biologically. And so we haven't quite got to genetics yet, but remember, this is what DNA does. So when we say that DNA and RNA, which is where we're going next, when we say that it codes for genetic information, that DNA in your chromosomes, all that's really doing is telling your cells what sequence of amino acids they're going to use to assemble proteins, right? And if you have a mutation, right, if your DNA gets changed to a slightly different sequence, and instead of, let's say, an aspartate amino acid that's supposed to be negatively charged and very water soluble, that you change that to a nonpolar isoleucine instead, that could disrupt the entire structure of the protein, change its function, probably usually in a negative way, and have huge implications for genetics. Okay, so amino acids build proteins. Proteins are really the functional units of a cell. That's how cells get things done. And so we care very much about what amino acids are used. When you're actually building a protein from amino acids, the chemistry is the same as it was for carbohydrates and lipids. We're still talking about using dehydration synthesis to connect them. And so in this graphic, this is amino acid number 1. This is amino acid number 2. And exactly like we saw in carbs, you'll take an OH group from one of them, you'll take a hydrogen from the other, and dehydration, because we are extracting water. They're not showing it here, but that H and OH are going to be removed, and you're going to get water as a waste product. so that the carbon from this amino acid and the nitrogen from the other are directly covalently linked to each other, right? That covalent bond between carbon and nitrogen that connects two amino acids, that's a peptide bond. And so if you look at the structure of an entire protein, and oftentimes it's drawn like this, where each little circle represents one amino acid, right? Glycine, isoleucine, valine, glutamine. They're not showing you the chemistry here, but every one of those pairs of amino acids would be linked by that peptide bond that we just saw on the previous page. Right. The other thing you'll notice is there are other ways to link proteins to each other. Only two of them are covalent, right? So within one chain, these are all peptide bonds. That's what we just talked about. If you're going to take two separate protein chains and link them together, you can use a type of bond called a disulfide. It gets its name because it's using sulfur atoms, and the only amino acid that even has sulfur is cysteine. And so if you want to make disulfides to link two chains to each other, the way that you are in this insulin protein, you'll need to use cysteines and covalently connect their sulfurs. Otherwise, all of the peptide bonds between amino acids, that is going to be the vast majority of the covalent bonds that you actually see in any protein. Okay. All right, so the next concept in this is once we have our chain of amino acids, right, and that chain of amino acids we refer to as primary structure, right? So if you get on an exam and you get asked a question about the primary structure of a protein, all that really means is the sequence of amino acids that make it up, right? After primary structure, things start getting a little bit more complicated because proteins can start to fold, right? And the folding we call secondary and tertiary structures. We'll see more details on this in a little bit. And so secondary and tertiary structure really just means taking that linear chain of amino acids and starting to fold it into a more complex three-dimensional shape, right? So this figure is showing you examples of secondary structures. In this coil, right, these molecules in the background, these are your amino acids, and they can form this twisted, almost like a spiral staircase structure. We call that spiral an alpha helix. They can also weave back and forth, forming alternating chains laying on top of each other, almost like folding a piece of paper with creases. Those are called beta strands. In a lot of cases, you'll also see these referred to as beta sheets or even beta pleated sheets because they have pleats or folds right between the chains. Okay, so these are examples of secondary structures, and important to remember, secondary structures are not held together by covalent bonds. They're held together by hydrogen bonds, right? If you don't remember hydrogen bonds, go back to a few videos back in the playlist. I've got an entire video talking about hydrogen bonding and how it happens, but this is just attraction between partially positive and negative regions of those amino acid structures, right? Once you have those secondary structures, you can take those and fold them back on each other to form tertiary structures, which really just means you're giving a three-dimensional shape to that protein, okay? And so primary structure means sequence of amino acids, right? 1, 2, 3, 4. Most proteins have hundreds of them. Secondary structures, alpha helices and beta sheets. Tertiary structures, folding those alpha helices and beta sheets back on each other to form an actual three-dimensional, usually globular structure. And then quaternary, the term quaternary structure means that you have more than one protein. And so in this example of hemoglobin, each of these different colored structures, 1, 2, 3, 4 of them, each one of these is a separate protein, but they stick to each other through some combination of hydrogen bonding or hydrophobic interactions. And so when two different proteins stick to each other and form this complex, then we call it quaternary structure, right? Fourth level. Okay, the biological significance of this is, like I said before, changing even just one amino acid in the primary structure. So in this example, we're looking at the gene that codes for hemoglobin. And in hemoglobin, amino acid number 6 is supposed to be a glutamic acid. In people that have sickle cell anemia, it's mutated into a valine, right? That one change in primary structure, switching around a single amino acid, can take the secondary and tertiary structures of hemoglobin and change it so that you get a defective shape in that protein. They don't fold correctly, and it's not going to function like it should. And so sickle-celled hemoglobin really just means that we have a change in a protein that's so small you can't even see it with a microscope. But when those chains assemble with each other, they don't assemble correctly, right? And you get mistaken interactions with other parts of the cell. And so in the end, you wind up seeing this, right? Normal red blood cells are supposed to be perfectly round and shaped, kind of like donuts. These guys are sickle cells because the shape of that hemoglobin alters how it interacts with the cytoskeletal proteins in that cell. And these sickle-shaped red blood cells don't flow as easily through blood vessels. They tend to clog and form blockages. And that's what causes the horrible cardiorespiratory pulmonary symptoms that you see in a sickle cell patient. And so the message to take away from this is, it may seem like a very small, trivial biochemistry thing, but any genetic disease, whether it's sickle cell or cystic fibrosis, or even like inherited Alzheimer's disease, if you zoom in close enough, all of them at some point are because of changes in protein function, because we just altered the amino acid sequence in that protein. All right. This is going to be really important later on in the class when we get to genetics. And so pay attention to this stuff. Remember your main points. Amino acids are monomers. They're used to assemble polymers made of proteins. And once you have those polymers made, we have to fold them using secondary, tertiary, and quaternary structures to get a final product that's functional. Okay? And remember, those sequences of amino acids are encoded by genetic information in your DNA and RNA. And that's a perfect lead into the next video that we're going to do, which is going to cover those nucleic acids. So follow that one next in the playlist. Make sure you subscribe so you get any updates, and we'll work through the rest of this chapter.